Okay, thank you very much for inviting me. I'm sorry that I cannot be there in person, but I'm really happy to be able to present our research virtually. One of the main research interests in my group is investigating genome organization using computational models, and it's a problem where computational chemistry has a lot to say. I hope I'll convince you in my slides. And this problem of genome organization is a field that has gained a lot of attention because genome sequencing today has become so efficient that it can now be used as a routing tool, which has actually enabled the challenge to be moved from reading our genome to trying to interpret the functions that it undertakes. However, this is a very hard problem because of different reasons. One reason is that our genome is huge. So if we were to take out the DNA in one of our cells and extract it out, we would see that it's over 2 meters long. Yet it has to condense dramatically in order to fit into the tiny nucleus of our cells that are almost 1 million times smaller. And this condensation process is very complex and also important. And just to put this in context, let me share with you this analogy that I really like about what more or less this problem entails. And the idea is, imagine that you take one of your hairs, a human hair, so imagine how thin that is. And now imagine that the hair is 20 kilometers long. And you try to compress it enormously in order to fit it inside a golf ball, so enormous condensation. And not only that, but the cells also face the challenge of trying to look for a precise region of your hair while the hair is still inside the golf ball. So it's quite a difficult problem. And not only that, but like folding the same piece of paper differently to obtain, let's say, a paper bird or a paper ball. Something similar happens with our genome, in which different ways of organizing the same DNA information in 3 dimensions are suggested to enable different functions. So in very simplistic terms, one way of organizing our genome may explain how cells become heart cells that beat, and a different way of organizing our genome explain how cells become neurons. And the question we are interested to answer in our group is how. So how these different ways of organizing the same DNA information emerge, and how is this organization shaped and maintained, and our focus is biophysical and molecular mechanisms. Now, these are really big picture questions. They are not exclusive to us. Many groups are interested in them, and they are attacking them from with different techniques, different perspectives. And the reason of this interest is that allowing us, solving these questions would allow us to understand how genome organization enables different functions during development or acquisition of cell identity, or also what happens when things go wrong in diseases or, let's say, aging. Now, in order to begin to understand these questions at the molecular level, let me quickly introduce to you the building blocks of genome organization, which are star players in all of our research. So if we were able to zoom in into this condensed genome that is known as chromatin, as I'm showing you here in my slide, and we would see first that the DNA is not found free in our cells. It's always complex with proteins. And this kind of makes sense. The DNA is a highly negatively charged polymer. So if you were trying to compress it to like extreme levels of condensation, you can imagine that you would face quite a challenge because of just electrostatic repulsion. So our DNA uses proteins known as histone proteins that are partially positively charged, and assembles these very important cylindrical nanoparticles that I'm showing you here that are known as nucleosomes. Nucleosomes are kind of like a smart glue that glues together and compresses all our genome. One of the reasons that it can do that is because each nucleosome has 10 intrinsically disordered protein regions. So very flexible, positive protein regions that extend out of the nucleosome core and kind of act like arms, allowing the nucleosomes to grab distant DNA regions, to grab other nucleosomes, to grab other proteins. So the way in which nucleosomes help in this selective condensation of the genome to enable different functions is via an additional chemical code on top of the genetic code known as the epigenome. So these nucleosomes carry different types of chemical modifications, including methylation, acetylation, phosphorylation, and recruit a wide range of additional proteins. And this chemical information is not distributed homogeneously across our genome, but forms kind of domains. So some regions are enriched in some chemistry and may give rise to active genes and active domains. And a different genomic region might be enriched in different chemical components and give rise to an inactive domain, a region where our genes are off. However, how this chemical diversity and genome function are interconnected is actually not well understood, and this is in part because we cannot see the organization of molecules inside these different domains. And And actually, at the moment, the structure of chromatin, this polymer, has mostly been resolved in two limits. One is the limit here that I'm showing you on the top left of small system sizes, but at really high resolution, atomistic or near atomistic resolution. And the other is the limit I'm showing you here at the bottom right, which is the limit of systems that are larger than the sizes of genes, and therefore they are functionally relevant, but with relatively low resolution. So we cannot see these molecules, as I was mentioning. And actually, these mechanisms of how the chemistry, the physical-chemical diversity of the genome gives rise to different functional domains happens right here at the blind spot that I'm showing you here. Now, at the moment, experimental techniques are advancing enormously, getting closer to the blind spot, which is very exciting. However, all these techniques actually rely on computational models to interpret the data and propose structures that match the experimental observables, which means that improving computer models is also really important, and that is what we are trying to do in my group. So we develop multi-scale modeling techniques that combine descriptions of different regions, different systems related to genome organization at different levels. So all the way from atomistic simulations of dinucleosomes to increasingly approximate coarse-grained models of different systems related to chromatin. And this allows us to get to really big system sizes, including phase-separated chromatin regions or big genomic regions, which I'll be talking to you about in a moment. Now, before I explain more about phase separation, I would like to first give you some ideas of how these polymers known as chromatin behave, so that then you can understand their properties and how they phase separate. And something that I want to mention is that for many, many years, experiments on chromatin were performed in vitro and in these like small polymers. And these experiments suggested that chromatin would fall into a very well-ordered structure, a 30 nanometer wide fiber, more or less. And therefore, the hypothesis was that chromatin inside cells also form this very ordered system. However, with experimental techniques, more recently have advanced to the point to see signatures of chromatin inside cells, and they have consistently struggled to find actually evidence for these 30 nanometer fibers, and have actually proposed this new model known as the liquid-like chromatin organization. This is not entirely surprising because in vitro experiments needed to be done with perfectly uniform chromatin, so all the nucleosomes being the same, all the DNA connecting nucleosomes also being the same. And actually, our previous model has demonstrated that if we treat chromatin as a homogeneous polymer, as a homopolymer, in which all the DNA is exactly the same, all the nucleosomes are the same, chromatin actually adopts this in vitro-like regular fiber structure that I'm showing you here on the left. However, if we consider variations in this DNA length, as occurs in vivo, we now observe a complete transformation of the structural landscape favoring polymorphism, as I'm showing you here on the right. And these results actually support the idea that considering heterogeneity, not only in the models, but also in the in vitro experimental systems used to assess chromatin organization is very, very important. We also know that the chemical composition of the histone proteins can completely transform the structure of chromatin, both directly and indirectly, by recruiting biomolecules. And when we look at the direct modulation of chromatin structure by chemical marks, one of the most dramatic changes is the decondensation of chromatin upon histone acetylation, lysine acetylation. This has been observed in vitro, as I'm showing you here in the slide, since like many, many years ago. However, the molecular mechanism of why this happened was not very clear. We combine atomistic simulations of proteins and dinucleosomes and discovered that acetylation induces a more complex effect than simple charge reduction. It is that it increases the stability of otherwise transient secondary structural elements in the histone tails, condensing them, making them rigid, and also unavailable to mediate internucleosome interactions. So when we map this significant rigidity into a coarse grain model, we found that actually this rigidification explains the unfolding, the decondensation of chromatin, while simple charge reduction didn't. Now, to try to get a little bit closer to mimicking, at least in part, this enormous complexity of physicochemical diversity of in vivo chromatin, we are developing coarse grain models that address a few points that we consider crucial. One is the chemical heterogeneity of the proteins and the DNA, the ability of proteins to target chromatin and transform its organization, and a key observation that has been largely overlooked, and is the idea that nucleosomes themselves are not rigid particles, they are actually quite plastic, deformable. And indeed, simple thermal fluctuations allow part of the DNA that is wrapping around the histone core to spontaneously unwrap and rewrap in this phenomenon that is known as nucleosome breathing. So nucleosome breathing is important, as I'll show you next. Now, my student Steve, shown here in the slide, developed a coarse grain model to accomplish considering all the features that I just mentioned. In his model, a few of the key ingredients is that all the amino acids are represented explicitly with one bit per amino acid, and all the DNA base pairs as well as are represented explicitly, and the sequence-dependent mechanical properties of the DNA are captured, modeled from the atomistic to the coarse grain model. Now, another important ingredient in our model is that the DNA binds to the histone core simply by electrostatic and hydrophobic interactions. So we are not forcing the DNA to be permanently bound to the histone core. And reassuringly, the model predictions are in quantitative agreement with mechanical unzipping experiments of nucleosomes, which means that these breathing motions are described in a reasonable way. Now, we can use this model to investigate the impact of nucleosome breathing on chromatin organization to see, does it make any difference to have this plasticity? So we do simulations with our models and measure in the resulting equilibrium configuration and ensembles the frequencies of interactions among first nearest neighbors, second nearest neighbors, and so on. And then classify these interactions as to whether the nucleosomes are perfectly aligned in what is known as face-to-face interaction. So this alignment that I'm showing you here at the top, or face-to-side interactions or side-to-side interactions, so just different orientations of the nucleosomes. And basically, when the nucleosomes are allowed to breathe, we see that there's a wide range of different interactions that they can engage in. So the important part here is that there's diversity in the types of interactions and also orientations. Now, in contrast, as a control case, what we did is now glue the DNA to the histone core. So now we are not allowing to have these breathing motions. So when we do that, what we see is exactly the same system, but now the DNA has no breathing motions. We now see that the chromatin formed is very well ordered, 30 nanometer fibers. All the DNA is, all the nucleosomes are mostly engaged with interactions with their second nearest neighbors, as you can see in this image as well. This is nucleosome 1, 2, 3, so 1 and 3 are always interacting. In addition, we can also monitor the dynamics of these nucleosome interactions simply by looking at how often two nucleosomes are bound to one another and represent this with a value of 1, or unbind and represent it with a value of 0, and then measure this across time for different nucleosomes in this system. So we see that in these 30 nanometer fibers, where the nucleosomes are not allowed to breathe, once two nucleosomes stack, they remain bound. They almost never unbind. In contrast, when nucleosomes are plastic, now they bind and unbind dynamically, consistently with this idea of the liquid-like chromatin organization. In addition, with our model, we can also get detailed information about the interactions that stabilize the different structural behaviors of chromatin. I'm not going to go into detail, but I'm just going to mention that these kind of rigid fibers are stabilized by face-to-face interactions with the histone cores, whereas the liquid-like behavior of chromatin is heavily contributed by the disorder histone tail. So protein disorder gives rise to a chromatin disorder as well, which I think is very interesting. Now, binding of disorder proteins to chromatin also disables stabilizes the rigid fibers and promotes liquid-like chromatin organization. Indeed, we did a study with a protein that is widely found in silent chromatin region, which is the H1 protein region. So first, with atomistic simulations of nucleosomes with this protein bound, we did atomistic bias exchange metadynamics. We confirmed that this the C-terminal tail of this protein actually remains disordered when the protein is bound to the chromatin. And then when we map this disorder into our coarse-grained models, we see that binding of this protein also gives rise to disorder liquid-like organization. So if we now start with one of these chromatins where we have inhibited deep breathing motions, we, as I mentioned before, see that it forms regular systems, and simply adding this disorder protein enhances the compaction and also transform the system again to this disorder type of liquid-like organization, where a wide range of nucleosome interactions are promoted. So in summary, so far what I've told you is that our simulations and also experimental work of our collaborators reveal that this fiber-like organization of chromatin, these regular systems, is actually very fragile. It needs regularity to emerge, and different sources of heterogeneity, including protein disorder, nucleosome breathing, favor liquid-like behavior. So this model that is known as liquid-like behavior, going by Kazu Maeshima. And this liquid-like chromatin organization actually challenges the idea that chromatin compaction unequivocally implies steric varying of enzymes, suggesting that some mechanisms, alternative mechanisms, need to be used to reconcile how function and structure of chromatin are linked. And this is what brought us to investigate phase separation, because actually one of the newest ideas that attempts to explain how chromatin might be organized in a space inside the nucleus too efficiently to control accessibility, and that doesn't rely in the picture that a rigid fiber-like structure is needed to explain the silencing, is this idea of liquid-liquid phase separation. The field of intranuclear phase separation has actually exploded in the past few years, and it has now been suggested that this mechanism has many potential functional implications. Some of the most famous examples is phase separation of a protein known as HP1, which is associated with the formation of silent chromatin heterochromatin domains. or the phase separation of transcriptional coactivators, which are suggested to enable the formation of super enhancer regions or transcriptional factors where genes are being transcribed. And not only that, but the Rosen lab showed that liquid-liquid phase separation is also an intrinsic property of just the chromatin polymers, and that without the aid of additional proteins, these arrays can undergo liquid-liquid phase separation. So go from a well-mixed state to a state where they have a condensed phase rich in chromatin in coexistence with a diluted liquid. So we were interested in investigating this phenomenon, and also we wanted to know if these breathing motions of nucleosomes actually play any role in phase separation, and for that we developed this multiscale technique. So combining, as I mentioned, atomistic simulations to develop a high-resolution chemically specific coarse-grained model of chromatin, and then from that model on, a minimal representation of chromatin, where now nucleosomes are represented with just a few particles. With this type of technique, we can model, we can simulate chromatin liquid-liquid phase separation, which is what you are looking at the moment, while we resolve individual nucleosomes and also consider these breathing motions as well. And one of the key quantities that you can measure with in phase separation is phase diagram. So this is a plot that I'm showing you here. In this particular case, is the space of salt concentration. It's inverse salt concentration. So it's go from high salt at the bottom to low salt at the top versus chromatin density. And basically we get this curve that is known as the coexistent plot, and it delimits the phase space of parameters that allow chromatin to phase separate. So all this shaded area below the curve is the set of parameters that promote phase separation, versus the area above the curve where no phase separation is expected to occur. So it's kind of what you are seeing in my slide, homogeneous system where everything is mixed versus a system where you have a cross-section of a liquid droplet at the center and the diluted liquid around it. So we can investigate how these coexistent regions are changed when we change the parameters of chromatin. That is something that we did. We started with a DNA linker length because our experimental collaborators have worked with that. So we wanted to see how well our model recapitulates their experiments. So in the experiment, the length of the DNA connecting successive nucleosomes was varied in this series 10n plus 5, so equal to 15, 25, and 35. And when we try those linker lengths, we see relatively big coexistent regions, which is consistent with the experiments of our collaborators seeing this formation of droplets very easily too. Now, when we try DNA lengths that are equal to 20 or 30 base pairs, now we see the coexistent regions become much smaller, which is consistent with no phase separation observed in the experiments. Now with simulations, we can go one step further and ask, why is this happening? And something that we do is we can do pooling experiments of single chromatin fibers with a different DNA length. And we see that the fibers that have a linker length of 10n are very hard to break, whereas the ones that have linkers of 10n plus 5 are actually very easy to break. And if we look at the patterns of interactions between these different types of systems, we see that because the DNA has a helical repeat of 10 base pairs, then the systems where the DNA has equal to 10 or 20 and so on, has nucleosomes that are perfectly aligned with one another, forming these actually rigid chromatin fibers. Whereas in the systems where the DNA is 10n plus 5, now we have this liquid-like chromatin organization where the fibers are way easier to unfold. And then we can also compute a potential of mean force from umbrella sampling simulations of two of these chromatin fibers, again with a different DNA length. And we see that these rigid fibers have relatively weak interactions with one another, whereas the liquid-like chromatin fiber, the ones with 10n+5 DNA lengths, actually interact with one another much more strongly, because when they meet each other, they kind of melt, engaging in a lot of interfiber interactions. So once we validated the model, we wanted to use it to investigate the impact of nucleosome breeding, as I mentioned, and consistently with what I just mentioned before, we also observed that breeding increases the size of the coexistent regions, of here in red. And again, this occurred because breeding motions promote this liquid-like behavior of chromatin, and inhibiting the breeding gives rise to these rigid fibers that do not want to bind to other fibers. A key quantity on phase separation is the valency. So the higher the valency, the easier it is for the system to phase separate. So when we compute the valency of nucleosomes, where nucleosomes are breeding versus non-breeding, we see that breeding motions promote the valency of chromatin, and actually they also enhance the network of connections between the condensed liquids. So that's why breeding is so important. Something that you can see quickly from my slide here is that actually nucleosome valency, the number of nucleosomes that each nucleosome binds to, is context specific. It varies with the salt concentration. And actually this variability of biomolecular valency with environmental conditions also emerged in a recent experimental collaboration that highlighted the strong modulation of protein phase separation by salt concentration. Quickly, I just mentioned that our experimental colleagues discovered that certain proteins can undergo salt-mediated reentrant phase separation. That means that they can phase separate both at low salt and high salt and be well mixed at intermediate values. So they re-enter the phase separation state at high salt. And our atomistic and coarse-grained simulations show that different molecular interactions stabilize the condensates in the low salt regime and in the high salt regime. So basically, at low salt, condensates are mostly stabilized by electrostatic cation pi and pi interactions, whereas in the high salt regime, they are stabilized mostly by pi and cation pi interactions. And also the valencies change, as I was mentioning. It's not valency, and the ability of systems to phase separate is not only intrinsically related to its chemical composition, but heavily modulated by the environment. Now, this also motivated us to think, can we design models to accurately predict the phase separation of proteins? And that is something that we did recently. We wanted to develop these models because they would allow us to screen the effects of sequence mutations with greater precision, rationally design biomolecular condensates with controlled properties, or characterize the material properties of these condensates. So in order to achieve that, we designed this model termed M-pi-pi, where each amino acid is represented by a single bit. All the bits are connected with harmonic springs, and the non-bonded interactions include electrostatics and short-range interactions. Now, in particular, to parameterize these non-bonded interactions, we use a multiscale approach that involves combining experimental data with high-resolution atomistic potential of mean force interactions to balance the contribution of the different interactions. Now, when we use this model when we try to test this model, we were able to use experimental data where that provided quantitative phase diagrams in the space of temperature versus protein concentration for a wide range of proteins, variants of the HNRLPA1 protein, which has served actually kind of like as a gold standard to test computer models to see how well they do. So when we test our model with this particular system, here I'm showing you the predictions of the model, the simulated critical temperature versus the experimental one. We see that our model performs really, really well and outperforms actually other similar models in predicting the critical parameters of this system. Once we have a model that performs really well in predicting the phase behavior of proteins, we can also couple it with genetic algorithms. For instance, if we want to rationally design mutations to enhance or inhibit the phase behavior of a system on purpose, that is something that we have done. And more recently, my student, Pindu, shown here, has been able to develop a genetic algorithm that goes slightly further, and the algorithm allows you to propose mixtures of proteins to promote or inhibit the formation of multiphasic condensates. So not all these condensates are homogeneous systems. Some have multiple coexisting phases within. And with her algorithm, you can actually decide, I want to have these two proteins and propose mutations so that the condensates go from homogeneous to unmixed, multi-phase. And with this type of approaches, we can also get physical-chemical descriptors that allow us to understand why condensates form these different types of structures. In particular, for instance, for multi-phase condensates, our techniques suggest that the difference in homotypic interactions between the proteins that you are mixing grows as you evolve them towards multiphasicity, and the heterotypic interactions weaken again as you evolve them towards multiphasicity. So in summary, I just wanted today to show you how we can use multi-scale models to complement experiments and link this subtle atomistic effect to large-scale organization of biomolecules and capture some of the complexity of these systems in vivo. Okay, so finally, I just want to thank my students, postdocs, for all their hard work, our collaborators, our funding, and thank you for listening.